Hello, and welcome back to Episode 6 of the NCERT Biology 365 podcast. Today, we are covering Section 8.5.2 and 8.5.3, the cell wall and the endomembrane system. If you are following along in your textbook, we are on page 94. We'll break down the chemistry of plant and algal cell walls, discuss why the endomembrane system operates as a single coordinated assembly line, and analyze the transport properties of the vacuolar tonoplast. Let's get started.
Let's start with the cell wall, which provides structural support and protection. NCERT makes a clear distinction between algal cell walls and those of higher plants. Algal cell walls are composed of cellulose, galactans, mannans, and minerals like calcium carbonate. Plants, however, utilize cellulose, hemicellulose, pectins, and proteins. This composition difference is a common target for matching questions in NEET, so make sure to highlight both lists in your book. Why is this called the endomembrane system? While each of these organelles is distinct in structure and function, they are grouped together because their functions are closely coordinated. The system includes the endoplasmic reticulum, the Golgi apparatus, lysosomes, and vacuoles. However, you must memorize the exclusions. Mitochondria, chloroplasts, and peroxisomes are not considered part of this system because their functions are not coordinated with the others. This is an easy target for choose the incorrect option questions. Discovered by Camillo Golgi in 1898, this organelle consists of flat, parallel sacs called cisternae, measuring 0.5 to 1.0 micrometers in diameter. The Golgi has a distinct polarity, a convex cyst or forming face that receives material from the ER, and a concave trans or maturing face that releases packaged vesicles. While structurally different, these faces are interconnected. The Golgi is also the primary site for the formation of glycoproteins and glycolipids. Finally, let's look at vacuoles. In plant cells, the vacuole is bound by a single membrane called the tonoplast, and it can occupy up to 90% of the cell volume. Here is the key transport concept. In plants, the tonoplast facilitates the transport of ions and other materials against concentration gradients into the vacuole. This active transport is why the concentration of these ions is significantly higher inside the vacuole than in the cytoplasm. Keep this active transport detail clear for your exams.
That concludes our look at the cell wall and the endomembrane system. To test your memory on these structural details, click the link and take our Day 6 CBT practice quiz. Also, don't miss our long video of Day 6. Join our Telegram group to download today's handwritten notes sheet, and we will see you tomorrow for Day 7 as we cover mitochondria and plastids.